Molecular machines display a key signature or hallmark of design, namely irreducible complexity. Molecular machines are the smallest irreducible forms of a machine, namely a structure which can perform a certain function. A motor uses an external energy to turn an axle. A switch cycles between two or more states. An actuator or shuttle can transport a structure from one end of a cable to another. All of these are seen in human engineering on a scale from kilometer to nanometer. Molecular machines are simply the smallest possible representation of these structures. An axle 1 atom thick. An on-off switch using a single bond. Synthetic nanotechnology is a recent invention for us humans, but nature has spent billions of years developing the architecture of the cell, and many of the proteins in our bodies have these same irreducible structures as the nanomachines we've synthesized in a lab. Why then have we spent so much time and effort making our own? The answer lies in the versatility and the scale of our own machines. But to understand the differences, we will need to take a closer look at these machines themselves. In this video, I will take us on a brief survey of the molecular shuttle, motor, and switch in some of their most well-known forms, both biological and synthetic. We will examine both the strengths and the drawbacks of each type, and I hope you'll come away from this presentation with a bit more knowledge of the field of molecular machines, as well as maybe a bit more wonder at the world around us. Our first machine is the shuttle. A linear motor, also known as a shuttle, is a machine that pulls itself along a linear structure, consuming energy in order to perform work. Many molecules in the natural world classify as shuttles, but there's no better example than the mighty kinesin. Kinesins are small motor proteins that walk along microtubules in cells to transport membrane-bound packages from one end of the cell to another. They get their characteristic swanky walk from the conformational changes of their protein subunits as ATP binds and unbinds to give the motor energy to perform work. The signature structure of a kinesin is its head, which has two binding sites which stick to their counterparts ATP and microtubule links via non-covalent interactions. ATP binding and ADP release changes the conformation of the microtubule binding site as well as the orientation of the neck linker. Together, these allow the kinesin to act as both motor and switch, alternating which leg walks forward with every other ATP. Well, yes, I know, a protein is technically a single molecule. But what if we need a properly molecule-sized walking shuttle? Well, one clever way would be to design a walker with 2 feet that can be bound and removed from the track with different conditions, allowing it to move forward and backward. A team at the University of Edinburgh did just that, binding 1 foot of their walker with a hydrozone and 1 with a disulfide bridge. Base unlocks the disulfide foot, and acid unlocks the hydrozone. But how can they make the walker directional? Kinesins do it by polarizing their microtubules so that the walkers can go electrically downhill, but ideally we'd want our walker to be able to go either way we chose. With some clever chemistry, the team designed their track using stilbeens, which are molecules that can change conformation from cis to trans using light. By controlling the shape of the track, the researchers were able to control the downhill direction of the track every time the walker lifted a foot, allowing the team to direct it either way they chose. Now, let's look at our second machine, the motor. A rotary motor is a machine which turns an axle, usually to power some device. Because they perform work, motors generally need to be powered by either a gradient or a direct force input, and there's no better example of this than ATP synthase. ATP synthase is a membrane protein embedded in the mitochondria, which catalyzes the formation of ATP. It works like a water wheel, channeling protons as they flow from high to low concentration in order to push its spin vanes around in a circle. As the axle spins, it uses that rotation to produce ATP every turn, which is used to power other things in the cell. To make a motor smaller than this, chemists have devised clever schemes to solve the problems of 1. Getting a molecule to rotate on command, and 2. Controlling its directionality. After all, motors don't tend to be very useful if they go forwards as often as they go backwards. One common type of motor uses catenanes, which are molecular chain links, where 2 rings are interconnected and fused so that they're inseparable. Thermal motion will cause them to wiggle around on their own, but unfortunately for the chemists, their average rotation will always net out to be 0. One team solved this problem by using 2 smaller rings locked into a bigger circular track, with unique binding sites along the length of the track. By shining specific wavelengths of light on the system, they could selectively change the binding sites from one which hydrogen bonds with the catenane ring into one which does not. This loosens the one ring to wander via thermal motion until it finds the next strong binding site to bind to, allowing the scientist to control its direction. And because of the unique two-ring system, the team found that they could use one ring, locked in place, to block the other, forcing the direction that each ring rotated every step. Now, let's look at our final class of machine, the switch. GPCRs are membrane proteins that stick out both above and below the lipid bilayer of a cell. On the top side, there's a binding site for signaling molecules. In the bottom, a different site which binds a different sort of signaler known as a G protein. The GPCR is finely tuned for a single signaling molecule which fits tightly in the top site. When this key finds its lock, the GPCR changes conformation, releasing the G protein below to go out and do cell things. Once it finishes, it comes back and binds to the GPCR, turning the switch back to its off position. In the lab, our switches can get much smaller. Rather than relying on 400 amino acids changing conformation, many of our molecular switches can switch on and off with just a single bond. Azabenzenes are one of the most common molecular switches. Their active site is just a single nitrogen-nitrogen double bond between two rings, which can undergo cis-trans isomerization when hit by a photon tuned to the frequency of their absorption band. This isomerization is one of the cleanest photoreactions known, with no byproducts, nearly infinite cycling capability, and reaction time in the picoseconds. Switches can also be made by making and breaking bonds. Spiropyrans, some of the first molecular switches to be made, work by making and breaking the carbon-oxygen bond of a pyran ring. While the closed form is colorless, non-planar, and mostly apolar, the open form is flat, polar, colorful, and binds metals and hydrogens well. This makes spiropyrans an excellent choice for controlled detection of heavy metals, pH, disease markers, and contaminants in both microenvironments and on a macro scale. So what do we make of this? There's clearly similarity between these biological and truly molecular machines. They clearly show the same irreducible complexity. But one is a single part of a larger integrated system, dizzyingly complex and almost unapproachable in its function. The other, atomically simple, able to be synthesized and customized, yet often made without any clear purpose in mind. Though these examples illustrate the same processes, they are not interchangeable in their function. Well, let's break it down in a table. Proteins only work in very specific conditions, generally around physiological pH and temperature, some up to around 100 degrees Celsius. Their power sources come from their environment and are similarly limited. Synthetic machines are simple enough to customize their tolerance to their purpose, so they can be made much more resilient and can be powered using just about any form of energy. Secondly, cellular machines are really good at doing one specific thing. ATP synthase can phosphorylate ADP madly fast, but adapting its active site to perform some other reaction is almost impossible due to the intricate nature of its assembly. In contrast, synthetic motors are often generic skeletal frameworks just waiting to be immured into any larger complex or anchored to a surface. On the other hand, synthetic machines are difficult to build up into a very complicated device. Even computer chips, which don't have any moving parts, still take hundreds of layers of material and billions of dollars in research to develop, and mechanical molecular machines with that level of complexity are still a long ways away. In contrast, biomolecular machines have had billions of years of evolution to adapt to and synergize with other machines within a cell, and so it's unfair to treat each one as a standalone machine. What makes a kinesin so impressive is not just that it can walk along a microtubule, but that it can execute complex commands with flexible cargo and do so in service of the entire factory. They can also regulate themselves in a way that our machines cannot, assembling themselves and often even healing injuries or misfolds without any intervention from us. Nothing that we can make comes even close. Ultimately, both types of machines have their use cases, and neither is going to make the other obsolete. A hammer is a terrible tool to paint a wall, as much as the paintbrush is for nailing it there in the first place. By learning more about every type of molecular machine available to us, we can get closer to solving the problems we're set to today, and developing the technologies of tomorrow.